So this part, we're going to look at the adaptive response. The adaptive response is all about lymphocytes. So we're going to be talking about T cells, T helper cells, cytotoxic T cells, but also B cells. Now B cells, when they differentiate into plasma cells, secrete antibodies. Those antibodies are antigen specific. The whole thing about the adaptive response is that it's antigen specific. Another key characteristic is that both T cells and B cells produce memory cells. And this gives us an immunological memory. This gives us an ongoing immune response. And of course, at the moment, in terms of our current situation with COVID, that is under a lot of discussion as to whether that's happening with COVID or not. In most cases it does. If it doesn't, if we don't have this immunological memory, then it's very difficult to make a vaccine. It's very difficult to have the disease and then be immune to it. So it's quite an exciting part, this. It's also something that is particular to us higher creatures, and is not common to earthworms or anything else. We have an adaptive response that gives us an ongoing immunity to something. Okay, so for a start off, let's get a bit of an overview of our adaptive, or you may see it called the acquired immune system. So we have, as you know from your Roskies, various lymph glands around the body. The thymus and the bone marrow are known as primary lymphoid organs. This is because they're involved in the production and or the selection of lymphocytes. So remember, lymphocytes are still leukocytes, but they're leukocytes that operate within the adaptive immune system. We have T cells. These are our cell-mediated immunity in terms of the adaptive response. And we have B cells that provide humoral or chemical immunity. That chemical we can call antibodies. When fully activated, T cells and B cells are specific to an antigen. Actually, to put it more accurately, they're specific to an epitope, a smaller part of an antigen. So we may have, for example, several antibodies that all will bind to different epitopes on one antigen. The other key thing about the adaptive response is that we have this memory. When activated, both T and B cells produce memory cells. Those memory cells are stored away for the next time we meet that antigen. So it gives us ongoing immunity. Okay, go back to the beginning. Immature T cells, they're synthesized, but then they have to go to school. They have to be educated. They're educated in the thymus gland. And this happens from sort of fetal age up to adolescence. The thymus then atrophies. And us adults, we produce very few new T cells. Most of our immunity is built up in childhood. So in the thymus, first of all, the immature T cells are presented with MHC by epithelial cells in the thymus. This is called positive selection. What it does, it tests that the T cells receptors on the T cells, logical name, you may agree, will in fact bind to MHC. If they can't bind, regardless of whatever is in that MHC, they will be useless. So therefore, T cells that won't recognize, that won't bind to MHC are, I'd like to say expelled, but actually they're killed. They're told to go into apoptosis and die. They're useless T cells. Then, still in the thymus, antigen-presenting cells present the immature T cells, the T cells being educated, with self-antigens from other body cells. So these are things we don't want the T cell to respond to. We want the T cell to tolerate these. And this is negative selection. What it does, it checks that the T cells will tolerate our own proteins and cells and not attack them. And as we know, sometimes this goes wrong later on or in this process, and we have autoimmunity where we develop T cells that will attack our own host proteins, but ideally not. If they do bind to self-antigens and react to them, then those T cells again are told to go kill themselves. They undergo apoptosis and die, because the last thing we want is a T cell coming out that is going to say, oh yes, I'll attack my own host proteins. And finally, our naive T cells, these are T cells that are still at school, babies, differentiate into either T helper cells or cytotoxic T cells, cytotoxic T lymphocytes, CTLs or TC sometimes. So T helper cells, they display a protein on their plasma membranes called CD4. And sometimes you may see T helper cells identified in text as CD4+. It means they have CD4, therefore they're T helper cells. Cytotoxic T lymphocytes display a protein on their plasma membranes called CD8. And they likewise may be identified as CD8+ in text. That just means it's a cytotoxic T cell. If you can't remember which one's which, I always find it quite helpful, of course, that CD4, the 4 is quite a spiky number, and H for helper is quite a spiky letter. In terms of cytotoxic T lymphocytes, CD8 is a nice curly number, and of course cytotoxic, the C, is a curly letter. It might help you to remember the two. The CD actually stands for cluster of differentiation, and we did come across it before in terms of the protection against membrane attack complex. We talked there of a CD protein. There's lots of different CD proteins. That one that protected cells against membrane attack complex was CD59, but there's quite a lot of them. They're part of this glycoprotein that sticks out of all cells, the glycocalyx. These are markers to identify cells. So our naive T cells, once they finish school, they leave the thymus. Once they leave the thymus, they are immunocompetent. In other words, they're functional in terms of responding to an antigen. They can recognize those antigens by binding to them. but they've not been activated against a specific antigen. B cells, on the other hand, don't go to school. They go directly to the tissues, usually from the liver or the marrow, prior to birth. So they go to the lymphoid tissues. They carry B cell receptors. These are actually like little antibodies, little immunoglobulins sticking out of them. Usually more than one, but that's as many as I could be bothered to draw. When activated, they differentiate into plasma cells. So being activated means that they become antigen specific. Once they become plasma cells, they secrete antibodies, immunoglobulins, as we're now going to call them. These immunoglobulins, the FV end, the two tops of the Y, are specific for the antigen that initially activated the B cell. Antibodies can circulate throughout the lymph and the blood. As we said, their B cell receptors are also antibodies, but of course these are mounted on the plasma membrane of the B cell, and of course they are antigen specific after that B cell has been activated. So let's have a look at the innate response through the different processes involved. We start with our antigen presenting cell. We have our antigen specific antigen presenting cell that then goes off and it activates specific T helper lymphocytes. These are naive but immunocompetent T cells within the lymphoid tissue. They bind to and they activate. This makes them antigen specific. T helper cells secrete interleukins, and they also help to activate cytotoxic T cells and B cells. So they really are helpers. They help to activate macrophages and lots of cells within the innate response and upregulate the whole response, primarily by secretion, but sometimes by binding directly, particularly with B cells. So we end up with the activation of specific cytotoxic T lymphocytes. So these are cytotoxic cells that will bind to specific antigen, the antigen that they have been activated against. We also get the activation of specific B lymphocytes, B cells, that will secrete antibodies that are antigen specific. Cytotoxic T cells cause apoptosis of specific pathogens and infected host cells. So their T cell receptor will bind to antigen and they will cause apoptosis of that target. B cells, as we said, differentiate into plasma cells. Plasma cells then secrete specific antibodies. Both of these, all of these, so T helper cells, cytotoxic T cells, B cells, will all produce memory cells. So some of the cells, half of the cells, when they proliferate, will go off and fight the current problem. The others will stay at home, will stay in the lymphoid tissue, and will be there for the next time we meet that specific antigen. So in other words, the next time we meet that specific antigen on subsequent exposures, we will have these memory cells stored away, ready to come out, and they're activated much more quickly. This is how we build immunity. So let's activate a T cell. As we said before, the link is the antigen presenting cell. This is our phagocyte that phagocytosed, and then it put some of the debris that it had phagocytosed, that it had broken down in its phagolysosome, into MHC and mounted it on its plasma membrane. So that antigen presenting cell then travels off to lymphoid tissue. It may not need to go that far. If we think in terms of skin-associated, mucosa-associated lymphoid tissue. So it goes off with that antigen MHC complex mounted on its surface. It finds a naive T cell that is able to bind to its type of antigen. So it might have to have a bit of a hunt around in the lymphoid tissue to say, oh, well, I've got, I don't know, a variant of influenza. Do you do influenza? Do you do viruses? Yeah, you do viruses. Okay, I can bind to you. No, says the naive T cell, I only do protozoa. So I'm not interested. So it might have to hunt around a bit, but it finds a T cell that's able to bind to its type of antigen. The APC binds to the T cell receptor via its antigen MHC complex. So the naive T cell has a T cell receptor, the antigen presenting cell has its MHC with its antigen mounted in it, in that notch at the top. They bind together. If it's a T helper cell, so CD4+, it will only bind to antigen that is presented on MHC2 complexes. The CD4, this protein sticking out, the cluster of differentiation protein, will also bind to the MHC. We need both of those things to happen. And then there's the binding of other proteins. So an example is something called B7 proteins on the antigen presenting cell bind to another CD protein on the T helper cell, CD28. And there's a few other things go on here that we don't really need to go into. The naive T helper cell that hasn't been made antigen specific before, it came out of the thymus gland, it's been hanging around the lymphoid tissue for ages waiting to be activated, it will now become antigen specific. So that little red blob in the MHC2 there on the APC will be the antigen that it will now respond to. Its T cell receptor will only now bind to that antigen. There are different subsets of T helper cell, and we're certainly not going to go into all of them. And what determines this really is that different interleukins may be produced by different subsets of T helper cells. So intracellular pathogens require T helper 1 cells. In atopy and hypersensitivity reactions, T helper 2 is the prevalent T helper cell. This is actually because atopic individuals tend to produce more interleukin 4, which differentiates T helper cells into this type of T helper 2, which then determines what interleukin that T helper cell will secrete. So there's different variants of T helper cells depending on the type of response and also the target, the nature of the target. So the subset of T helper cell then secretes different interleukins and interferons required to resolve the specific stimulus of the immune response. So you might require different interleukins and interferons, for example, for a viral attack, as you might for a protozoal attack, or as you might for some sort of tissue damage. So an example there is interferon gamma, IFN gamma, protects against intracellular pathogens. It protects against primarily actually viruses, which tend to be intracellular pathogens. T helper cells can even become anti-inflammatory. These anti-inflammatory T cells are known as Treg, and we'll come back to these particularly when we talk about GI immune mechanisms in clinical reasoning. Treg secrete interleukin 10. Interleukin 10 is anti-inflammatory. So what we're saying is that on activation of T helper cells, interleukins are secreted by the antigen presenting cell and then by the T helper cell. Both of them bind to the T helper cell and promote T helper cell differentiation. T helper 1 is our standard response, really. Our most common response will be T helper 1. So cytotoxic T lymphocytes are activated in the same way, but in this case it involves antigen presented on MHC1. So our cytotoxic T lymphocytes, CD8+, will only bind to antigen MHC1 complexes, not 2. And again, we still have the co-binding of CD proteins, but in this case, of course, cytotoxic T lymphocytes have CD8. So CD8 also binds to the MHC1. We also have the binding of other proteins, so we have B7 proteins binding to CD28, etc. The T cell receptor on our cytotoxic T lymphocyte will now be antigen specific. It will now only respond to the antigen that was presented by that antigen presenting cell on MHC1. Remember that all cells have MHC1, including immune cells. This binding in terms of T helper cells and cytotoxic T cells of the additional CD and B7 proteins is known as co-stimulation. If it doesn't happen for any reason, the T cell will go into anergy. It will tolerate the antigen rather than reacting against it. So we will lose our opportunity of developing long-term immunity. So what do? We've activated them. What do they then do? So let's start with T helper cells, CD4+, and see what they do. They activate macrophages and dendritic cells through the secretion of interferon gamma, but also via direct binding via another cluster of differentiation protein, CD40, on the T helper cell and the phagocyte. So either by direct binding or via the secretion of interferon gamma, they activate macrophages and dendritic cells that of course operate in the innate response. So they upregulate the innate inflammatory response. They secrete interleukins depending upon the type of T helper cell, whether it's 1, 2 or whatever. They will secrete interleukins of various kinds, and there's about 30 odd interleukins. They will also secrete other mediators. Those interleukins and other mediators stimulate leukocytosis. So here you can see we've got upregulation again. The expression of cell adhesion molecules on endothelial cells, so that then assists immigration of the leukocytes, and chemotaxis, which is guiding those leukocytes once they're into the tissues to the target area. CD4 plus T helper cells also upregulate cytotoxic T cells, and they do this by causing dendritic cells to secrete more co-stimulatory interleukins and by enhancing viral antigen presentation in infected cells so that the cytotoxic T cells can then bind to its specific antigen that's presented. T helper cells also bind to B cells, displaying an antigen MHC complex, and this helps to complete the activation of B cells. And this T cell dependent B cell activation is vital to getting long-term immunity and long-term antibodies or effective antibodies. CD8 cytotoxic T lymphocytes, as I think we intimated before, work a bit like natural killer cells, but of course they're antigen specific this time. They don't care whether the cells have MHC. What they care about is what's being mounted on that MHC. So cytotoxic T lymphocytes leave the lymph system, so they disappear off into the circulation, and they seek specific targets, specific in terms of antigen specific. And they are key really in terms of eliminating infected or malignant cells. This is their primary role. More recently, they have been shown that they can directly kill extracellular pathogens. But most of the papers that you'll see will talk about TC cells, cytotoxic T lymphocytes, in terms of infected or cancer cells. Their best known function involves an action similar, as we said, to natural killer cells, the difference being that they are antigen specific. So our cytotoxic T lymphocyte binds via its antigen specific T cell receptor to antigen presented by MHC1. This isn't activation. This is once it's been activated, it's gone off into the circulation, it finds some poor little cell that has been infected, or it's damaged, it's unwell, and on its MHC1 it's displayed an unhappy flag. It's displayed some damaged proteins or some viral proteins to say, I'm infected or I'm damaged, I'm ill. So if that cytotoxic T cell is specific to that antigen, it will bind to it via its T cell receptor. The cytotoxic T cell secretes, remember, perforins and granzymes? Yeah, this is where we have the similarity with natural killer cells. Remember, granzymes are proteases, and remember what they do to target cells. The target cell then goes into apoptosis, which of course, if it's an intracellular pathogen, it seals it within those apoptotic bodies so that it can then be efferocytosed by phagocytes and completely destroyed. It's not released to go off and infect other cells. So the apoptotic bodies with their little pathogen in are then efferocytosed. If this is a malignant cell, of course, it gets rid of that malignant cell and it's unable to replicate and develop into a full-blown tumour. So B cells. Now B cells can be activated in different ways with different effects. Most often, and this is what we want, this is the strongest response, this involves T helper cells. This is T cell dependent B cell activation. So in this, what happens is antigen binds to the B cell receptor, and remember we said that B cells could be phagocytes. So they're able to phagocytose, just like macrophages and dendritic cells. So the B cells phagocytose the antigen. The antigen is then presented, so in other words, our B cell is acting like an antigen presenting cell because it then presents the antigen via MHC2 on its surface. So far it's acting just like a macrophage or a dendritic cell that becomes an antigen presenting cell. It, like other antigen presenting cells, then travels off to the lymphoid tissue, and what happens is an antigen specific T helper cell. So this T helper cell is already activated against that antigen, binds via its T cell receptor to the MHC antigen complex. So it's already been activated. What it's doing is binding to a B cell, binding to its specific antigen presented by a B cell. And that fully activates the B cell. This is the best, strongest reaction that you can get from a B cell. This will give you memory cells, this will give you strong, long-lasting antibodies. So the B cell receptor born by that B cell is now antigen specific. And the B cell can become a plasma cell and it can secrete antigen specific antibodies. So this is T cell dependent B cell activation is the best possible response in terms of B cells and the production of antibodies. And then we have T cell independent B cell activation. So here we have something like a polysaccharide or a lipid, something that's not based in protein, binding repeatedly to the B cell receptor. So one binding doesn't do it, you need repeated bindings. This gives a weaker response of shorter duration, and it doesn't produce memory B cells. So we are not going to have an immunological memory in terms of the production of antibodies. And as we suggested before, this has become quite significant in terms of COVID, because it has been thought that this is what happens, and hence we have a very short immunity to COVID. So what do antibodies do? Well, they're immunoglobulins, and we'll call them immunoglobulins. As we said, they are Y-shaped. They are actually glycoproteins, so we have a sugar and a protein. And this is their monomer state. This is their normal individual state. So this might be IgG, which is the normal subsequent exposure antibody. It might be IgE. If you remember, IgE was the one that bound to mast cells in allergic reactions. But these monomers, if you stick them together, they can form other classes of immunoglobulins. More importantly, there we have IgM. Look at IgM. Count how many fragment variable bits we have. If we're thinking about the monomer, the top of the Y, we have two fragment variable areas, two areas to which antigen can bind. If we look at a pentamer such as IgM, we have 5 of those monomers together, therefore we have 10 fragment variable areas that can bind antigen. And we also have a dimer there. This is IgA. We'll come back to IgA in terms of the GI tract in clinical reasoning. It's a dimer. We've got 2 monomers stuck together by a little joining protein in the middle. So the antigen binding sites at the top of the Y are called fragment variable, or FV for short, because they are antigen specific. This is where the antigen will bind. The lower region is fragment constant, or FC. So this is where they bind to FC receptors. So they can bind to cells or complement to activate it, bind to cells in terms of phagocytes in opsonization, and it also helps to denote the Ig class. So it may differentiate IgG from IgE, both of which are monomers. On first activation of our B cells, so if we have a naive B cell, it meets an antigen for the first time. The B cells will display on their surface and secrete IgM. That's the one, that's the pentamer with 10 FVNs. On subsequent exposures, B cells will generally display IgD, which is another monomer, as their B cell receptor, and they will secrete IgG. That is our standard antibody, really, once we've been exposed to something for the first time. As we said, when activated, our B cells differentiate into effector plasma cells. These plasma cells, so the B cells become bigger. They go back into the lymph and the blood, and they start secreting antigen-specific immunoglobulins. So in other words, these immunoglobulins on first exposure to an antigen will be IgM, on subsequent exposures will be IgG, but the FV section will be antigen specific. Whatever the antigen that activated that B cell, those antibodies, the FV, will be specific for that antigen. So what do antibodies do? Well, one key thing is agglutination. So this is where each FV region can bind to an antigen. So if we have an IgG, it's a monomer, it only has 2 FV regions. But if we have an IgM with 5 monomers, it's a pentamer, then it has 10 FV regions. So potentially it can bind 10 separate antigens. What this does is it clumps the antigens together. So when the Fc region binds to Fc receptors on phagocytes, for example, multiple antigens can be phagocytosed together. So that phagosome will encompass potentially 10 antigens rather than 2. Opsonization we've already talked about. So when antibodies bind to the antigen via the FV region, that Fc region can bind to Fc receptors on phagocytes, which of course triggers phagocytosis. So, okay, if we have a monomer that may only be 2 targets, but they're still being phagocytosed. This is opsonization. If you remember, our other opsonin was C3b, one of our complement proteins. Neutralization. Now this is where the FV region binds to pathogens or to toxins secreted by pathogens, and by binding to it, it stops them binding to target host cells. So pathogens will often use existing receptors on target cells. So if you think of SARS-CoV-2, that uses ACE2 receptors, angiotensin converting enzyme 2 receptors, and it binds to them to get into the cells. It's a bit cheeky, but that's what it does. If you stick an antibody on that, it will stop it binding to the target cell. So it prevents the virus binding to host cells, which prevents its endocytosis and prevents its replication, because viruses need host cells to be able to replicate. Another thing neutralization can do, even if the virus can get into the cell, is to prevent that virus from uncoating. It needs to uncoat to release its RNA or DNA to be able to replicate. If you can stop it uncoating, it can't do that, it can't replicate. Another thing, if you remember when we talked about complement, we talked about how it's activated. And this is where antibodies can also come in. So the FV region binds to the antigen. Inactive circulating C1 from the liver can then bind to the Fc region. And what this does is it activates the C1. This C1 then goes through the alternative pathway and the classical pathway, and we end up with C3a, C3b, C5a, and then C5b through to C9, which of course are membrane attack complex. And most leukocytes do have FCRs, receptors for the Fc region of an antibody. And what happens when an antibody binds to them is that those leukocytes become activated. So they start doing whatever they do best, whether that be secreting inflammatory mediators or phagocytosing or whatever. So antibodies can help reinforce, if you like, the innate response as well. Okay, so another role of antibodies. Remember on natural killer cells, we talked about them before in terms of looking for host cell MHC. And we talked about them operating within the innate response. In this case, antibodies can help make them antigen specific. This is antibody-dependent cell-mediated cellular cytotoxicity, ADCs for short. So our FV region, fragment variable region of the antibody, this is the antigen-specific region, binds to antigen, whether that be on an infected cell displayed on MHC, or whether it be on the wall of a pathogen. Along comes a natural killer cell, and it has an Fc receptor. That Fc receptor can bind to the Fc region of the antibody. The natural killer cell then does what it does best. It releases perforins and granzymes. The infected cell or pathogen then goes into apoptosis and dies. So, to look at our antibody functions, we have agglutination where we're looking at multiple FVs binding multiple antigens. Opsonization where we're looking at antibodies binding to potential targets via the FV and then binding to an Fc receptor on a phagocyte. Neutralization, where we're looking at antibodies binding to a target and stop it binding to our host cells and going intracellular. In terms of viruses, this can prevent their replication. The activation of complement, we have antibodies binding to a target while the Fc binds to C1. C1 is then activated and we get our whole range of complement proteins that degranulate mast cells, opsonize, or form membrane attack complex. Antibodies can also upregulate inflammation by binding to Fc receptors on innate leukocytes and stimulating those. And finally, we have antibody-dependent cell-mediated cytotoxicity, ADCC, where we have antibodies turning natural killer cells into antigen-specific natural killer cells. Another key aspect that we intimated earlier on about the adaptive response is this ability to provide memory. So antigen-specific T and B cells, so they've been activated, they have been made antigen-specific by whatever antigen bound to them via MHC. Once they're activated, they proliferate, so we get more of them. Part of those are antigen-specific effector, T helper and cytotoxic T lymphocytes, or B cells that become plasma cells. So they go off and do the business. but we also, some of those cells that are proliferated will remain in the lymphoid tissue for subsequent exposure to that antigen. So we will build up an immunological memory. So both T and B cells make memory cells. These are stored in the lymph gland and the tissues. So our first exposure is a bit slower because we have to activate naive T cells and B cells. At that first exposure, though, we create memory cells. Memory cells that can come out an awful lot quicker. They've already been made antigen specific to fight any problem. And they increase with every subsequent exposure. So memory cells are stored in lymph glands and lymphoid tissue, or they circulate around for a while looking for any more potential targets that they are specific for. And some T cells actually become resident in tissues, a bit like our wandering versus tissue macrophages. They lodge in skin, lungs, bone marrow, intestines, anywhere that is a potential for invasion. And they're there ready to be activated as soon as that antigen is met again. So memory cells are activated with less intense stimulation. So we need fewer MHC antigen interactions and less co-stimulation. This makes their reactivation on subsequent exposure much faster. So responses on second and subsequent exposures are faster, stronger, and more persistent. And I'm sure you've had days where you think, oh, I think I'm going down with something, I feel really unwell, da-di-da-di-da. And it lasts 24 hours and the next day you feel absolutely fine. This is where your adaptive immune response has kicked in and dealt with the problem very, very quickly. So the next day, you're feeling great. It's a bit ironic that it's usually a day off that you're feeling rough and then on a work day you're feeling fine. But this is the concept behind this development of immunity, whether that be natural immunity due to being exposed to it or whether it be artificial through immunization. So immunity may be passive, so this is antibodies from maternal milk or injected immunoglobulins. Now immunoglobulins, of course, what we're doing is we're giving someone the antibodies. We're not necessarily stimulating memory cells. It can also be active where we have an immune response to natural antigens or to medical vaccines. If you give a vaccination, then what you're doing is you're stimulating the T cells and the B cells to produce memory cells that will fight that target in future. So let's look at that in terms of a couple of exposures. So this is our first exposure to antigen A. So we've got a moderate response. It's taken a little while. At the bottom we've got time in days. So 6 days and we're starting to build up a bit of response from the adaptive immune system. Of course, the innate immune system will have been working throughout, but this is adaptive. This is measured by the level of antibodies. So antibodies come out, they deal with it, and that then resolves. The second part, if you look at the blue line again, we've got second exposure to antigen A. And look how bigger that is, but also not how bigger it is, but how persistent it is. It's a much stronger response in terms of speed and also duration. But meanwhile, pink line, we've been exposed to our antigen B. So we have to develop a whole new set of antibodies to B. So our first exposure of B is moderate, as it was to A, hence the specificity. Of course, our next exposure to B will be a bit like our blue line there. It'll be much faster and much stronger, more persistent. So this is how we build immunity, through the development of these antigen-specific memory cells. Okay, final word on repair. So hopefully we've got it all settled down. We've removed the triggers and we've removed the problem, got rid of the infection, or got rid of whatever it was that was damaging our tissues. And we've got a situation where fibrin and clotting, so fibrin, we might have fibroblasts secreting collagen, we'll have clotting. This is walling off the damaged area so it can't spread. As we said, we've got fibroblasts. They're secreting extracellular matrix and collagen. This is filling in any gaps where we've got the death of cells until such time as we can proliferate cells into that area. Myofibroblasts are fantastic. Myofibroblasts are a type of fibroblast and they secrete very elastic collagen. And this elastic collagen helps to draw edges of damaged tissue together. In some people, they're a bit overactive. And this is where you get the keloid type scarring. If you've ever seen a keloid type scar, you've got lines across it. And this is where the myofibroblasts have pulled together a little bit too tight. They've gone a bit mad. So we have fibroblasts trying to reduce the area, build a scaffolding for new cells to grow into. Fibroblasts also secrete something called fibroblast growth factor. Endothelial cells secrete something called vascular endothelial growth factor, which encourages blood vessel growth into the healing area. Fibroblast growth factor promotes cell replication and tissue regeneration. So it will stimulate that cells, the existing cells, to go into mitosis and fill the area up. And then we have our vascular growth factors, like vascular endothelial growth factor. They promote something called angiogenesis to support cell proliferation. This is the budding of existing capillaries into damaged tissue to provide a blood supply. These little buds from the capillaries are very delicate, and this is where, if you've ever done any wound care and used to scrub it with a bit of gauze, and you'd get these little bleeds in the base of the wound, what you're doing is knocking off the top of these little budding capillaries. They're trying to grow into the area to support the new growth to fill that area. And then we have MMPs. These are proteases. It could be called proteinases or proteases. Matrix metalloproteases, MMPs, are really useful because we have our fibroblasts laying down this extracellular matrix for the cells to proliferate into. But of course, at some point, you have to remove the extracellular matrix to allow those cells to move into the area. So they have to be very, very carefully balanced, these MMPs. And they're actually balanced by tissue inhibitors of matrix metalloproteinases, TIMPs, to ensure that they only break down as much as is needed to support cell proliferation. So even repair is quite a complex process. However, if too much tissue is lost, or if the cells can't regenerate, so particularly if they're not epithelial cells, for example, maybe they're neurons or something like that that don't proliferate very well, then that fibrosis can become permanent. So the collagen that the fibroblasts lay down becomes the permanent architecture. And of course, that's invariably dysfunctional. If it's in muscle tissue, it doesn't stretch. If it's in an organ, it doesn't function as the cells within that organ would function. So we've lost some tissue and replaced it just really with a bit of packing. So that was the end of immunology. There's a lot to learn in terms of immunology, I know, but it is so massive. It is so important in terms of so many different conditions. There is probably no condition you can mention that doesn't involve some element of an immune response. So it is very, very important that you get this subject, particularly before clinical reasoning, under your belt. And there is an Amando's overview to help you, so please have a look at that. I'll put the link on Moodle for you and you can have a look at that. Most of it is fairly straightforward. It's just a lot of chemicals, a lot of cells all interacting together at once. And if you can get your head around that, then you'll be doing really well.